So yeah, Chris is a tough act to follow, but I mean, his story is really the inspiration and what motivates me to do basic science research to try and make, everybody, unfortunately, if you do have a stroke, more like Chris and live a full, long and prosperous life. So I'll tell you a little bit about the research I'm doing in my laboratory at University of Victoria in medical sciences. And my laboratory, one of our strengths is using imaging technologies to see things in mouse models of stroke that you could never see in humans. So I'm looking at things that are on the order of 1 micron, which is, for example, that's 1/1000th of a millimeter. So picture a millimeter and then divide that by 1,000. And these are the structures that allow your brain to function, really, for brain cells to talk to one another. So on this slide here, this is a neuron or brain cell. And you see this is the cell body. It has all these beautiful processes coming off. And then these little buds here. And these little buds are where brain cells talk to one another. And so I study how these little nubs, like leaves on a tree, change after a stroke. But I can also monitor blood flow at the same time and relate changes in brain structure with interruptions in blood flow. So let's go to the next slide. I won't really spend much time in this one since Paul introduced some of the basic statistics on stroke. But of course, it's a huge problem in society, and there's no cure. There is some treatments that help, but we still have a long way to go. And hence why I think research is vitally important to advancing treatment on stroke. So how do we model stroke in the lab? So I work with little mice, and essentially what we do is we use this model where we produce a stroke in the forelimb somatosensory cortex. So mice, just like humans, have each part of their body is mapped onto their brain, and a certain part of your brain processes information from your hands or your feet. And so what we do is we would produce a stroke in the somatosensory cortex that processes information from the limb, and then so we can track how the animal uses the paw after the stroke, as well as correlate that with changes that go on in the brain and sort of see what kind of patterns are associated with, for example, better recovery of paw use after the stroke. So the way we do this is we inject a photosensitive dye, and then we shine a laser over the skull, and then what that does is it causes clot formation, much like an ischemic stroke or a clot-related stroke, which is 75%, 80% of all strokes are ischemic. And so you get this area of death, and we use these mice. So this is a picture here. This is what the brain looks like about a week after the stroke. So it's quite obvious to see where the stroke is. You see this sort of swath of dead tissue. But we take advantage of these technologies, genetic approaches, where these mice express fluorescent proteins that float around in the cell bodies of neurons, of these brain cells. And so what's beautiful about this is that you can put these mice under a microscope, and because their brain is glowing and you can see the structure, you can actually watch how the brain changes over time. So it's really advanced. The field of neuroscience is using these mice that have neurons, brain cells that are already labeled with fluorescent proteins. So this is just a quick movie of showing you sort of as my old professor used to call it, the car accident, and watching a car accident as it's happening. So here's a picture of the surface of the brain, and we injected the dye, and we photoactivate. So we're going to activate the dye and activate the clots in that boxed region there. And so you'll see what happens to blood flow very quickly after clots form. And so you can see you'll see the blood flowing here through these arteries here, and then very shortly after turning on the green laser, the green light, you'll start to see blood flow slows down. There was a clot that just moved through. And so as soon as you can start seeing individual blood cells moving through the vessels, you know that there's a severe restriction in blood flow. So this mouse at this point is having a stroke, an ischemic stroke. Actually what happens is it's like plumbing. If the ends of the tubes get blocked, actually the flow goes in the opposite direction, which looks like it's going on here. what happens also is that there's waves of activity that propagate from the damage. And they start near the damage and then they radiate outwards here. And this is, in scientific terms, called spreading depression. But it's like a tsunami of electrical activity that's radiating outwards from where the stroke occurred. And this is actually really a clinically important problem, because even though you may be a brain cell far away from the stroke that survived the initial damage, these waves of electrical activity can actually further or jeopardize the health of those cells that survived the initial insult. So we're starting to learn about these propagating waves that go on long after the stroke, and they can go on for even a day or two after the stroke. So if you look at, again, I mentioned you can image structure and blood flow at the same time. So using these mice, you see this is part of a dendrite, which is the sort of a process of a brain cell, and you see these little leaves, which are where the neurons, brain cells talk to one another, and this is a blood vessel in red. And so this is before stroke, and then within about 10 minutes of forming a clot, so this is a clot here, so the blood flow is stopped to the brain. Within 10 minutes, you can see that these processes start to look a little bit off. Within 30 minutes, these neurons start to look like beads, and you can see it just gets worse over the 3-hour period. So these changes in brain cells occur very rapidly when you have a stroke. And again, this is why we emphasize that time is brain when you're having a stroke. So it is important, obviously, if you are showing signs, to get treatment. But this is a really amazing, I'll actually should go back here just to make sure. This next slide here is from my old supervisor, and he wanted to know if cells look like this, is there any hope? Can we restore the structure and integrity of the brain cells if you restore blood flow? Because one of the only treatments that works for humans is busting up clots and restoring blood flow. And so he occluded blood flow and was imaging brain structure here. So this is before the stroke. And then within 4 minutes of blocking blood flow, you see the structure of the neurons as pretty much disintegrated. But then you let the blood reperfuse. And within 10 minutes, things start looking better. 17 minutes, you can see structure is almost returned completely to normal. So the brain is resilient if you can restore blood quick enough. Of course, you can't go without glucose and oxygen too long, or there's no way. It doesn't matter, what you do after a certain period of time. So, and then I've also looked at a little bit beyond the first couple hours after a stroke, and this here is, again, this is just showing you a picture of what a stroke looks like. This is 6 hours after inducing a stroke, so there's already quite this, again, a lot of dead tissue or dying tissue. And if you look at some of these brain cells in the center of this area, you can see that they've lost all those beautiful processes. It's like they're being eaten away. But then if you look even in the border of the stroke, so if you look out here, what's really interesting is that the damage it evolves over time. So when neurons start to die, they start to have these little beads, right, like you saw earlier. And so this is not quite a healthy, but it's a surviving neuron around the edge of the stroke that looks like it's slowly being degraded, as you can see here. So, and what happens over time, especially over the first 24 hours, these are those little where the connections occur. You start losing those connections. There's about a 25-30% loss of those connections in the first 24 hours, even for cells that, for example, do survive the stroke. So you can see there's a lot of challenges the brain must overcome when it has a stroke. And of course, not only if the brain cell survives, it still loses tons of connections. So it has to remake these connections. So of course, if you've had a stroke, and I don't think we'll ever completely prevent stroke or damage, but we need to also know what to do with the brain after the stroke to sort of make best use of what's left. And so this is what's kind of fascinated me for the last 5 years, is trying to understand these changes that go on in the surviving regions of the brain. So in this case, I'm assuming we've lost part of the mouse brain here, but we're going to try and see what's going on here in the surviving areas that might be responsible for some recovery of function, like how Chris experienced it. So one of the ways we do this is you can't ask a mouse, how it's doing, so we observe them. And we use a number of tests. For one, this one here is a cylinder test. So we look at, after a stroke, does the mouse, when it rears up against the walls of a cylinder, does it use its left paw? So we always induce a stroke in the right hemisphere. So after a stroke, it doesn't use its left paw. It kind of tucks it in like this when it rears up against the walls. But over time, the mouse actually starts to use both paws again with some symmetry. We can also have mice reach for pellets. This is actually a rat, but it was a good image from a paper.